I'm Denis David, a professor of pharmacology, co-director of the Mood teams, and deputy director of research at the Graduate School of Health and Drug Sciences. In this month's webinar, I will be discussing the role of adult hippocampal neurogenesis in the response to antidepressants. Adult hippocampal neurogenesis is a unique and exceptional process in the mammalian brain which generates new neurons. Adult hippocampal neurogenesis is limited to only one part of the hippocampal formation, the dentate gyrus. The hippocampus is one of the several brain regions that together comprise the hippocampal formation. The hippocampus proper consists of four subfields, CA1, CA2, CA3, and CA4. The other region that together includes the hippocampal formation consists of the dentate gyrus, the subicular complex, and the entorhinal cortex. The three synaptic loops include the projection from the dentate gyrus to the cornu harmonious CA3 region via the Mossy fiber pathway, the projection from CA3 to the CA1 region through the Schaffer collateral pathway, and finally the projection from CA1 to the entorhinal cortex. The hippocampus is part of the limbic system, a group of structures involved in regulating emotion, learning, and motivation. Adult hippocampal neurogenesis refers to the production in the hippocampus in the post-embryonic stage of a single neuron type, the granule cell of the dentate gyrus. These granule cells are the main excitatory neurons of the dentate gyrus. Adult neurogenesis occurs in the dentate gyrus because of a radial glial-like neural stem cell pool. Adult hippocampal neurogenesis involves several steps, and more than 4 weeks are necessary to generate mature granule cells. The gradual transition in marker expression for each hippocampal stage is represented in this cartoon with a color-coded bar, with in red the proliferation stage, in blue-purple the survival maturation stage, and in green the mature stage. Neurogenesis is a form of plasticity that is unique to the dentate gyrus. Exercise with environmental enrichment increase adult hippocampal neurogenesis, while social stress and glucocorticoids decrease it, suggesting that life experiences dictate how adult-born neurons contribute to hippocampal structure and function. Classical monoaminergic antidepressant, electroconvulsive seizure, or even enriched environment increase monoaminergic neurotransmission. This increase in monoaminergic neurotransmission increase BDNF and stimulate adult hippocampal neurogenesis. To assess whether adult neurogenesis is required for the antidepressant mediated reversal of chronic stress treatment, we submitted an animal to focal hippocampal exirradiation prior to a chronic corticosteroid regimen, alone or in combination with fluoxetine. In corticosteroid-treated mice, where hippocampal neurogenesis is abolished by X-irradiation, the efficacy of fluoxetine is blocked in some, but not all, behavioral paradigms, suggesting both neurogenesis-dependent and independent mechanism of antidepressant action. Recently, we investigated the mechanism underlying the effect of antidepressant fluoxetine on behavior and adult hippocampal neurogenesis. After confirming our earlier report that the signaling molecule beta-arrestin 2 is required for the antidepressant-like effect of fluoxetine, we found that double cortin expression can be increased even though adult hippocampal neurogenesis is not. We discovered two other conditions where a complex relationship occurs between the number of double cortin expressing cells compared to level of adult hippocampal neurogenesis. A chronic antidepressant model where double cortin is upregulated, and an inflammation model where double cortin is downregulated. We conclude that assessing the number of double cortin expressing cells alone to quantify level of adult hippocampal neurogenesis can be complex, and that caution should be applied when label retention techniques are unavailable. So if you are interested on the field of adult hippocampal neurogenesis, you can find more publications from the Mood teams on our website. Thank you.